Welcome. Today I will explain the basic structure of cryptochrome and its various associated photomorphogenetic effects. As we all know, that cryptochrome, which is a blue light photoreceptor of plant, and they are also found in various insects, animals, etc. The chromophore of the cryptochrome formed by the flavin, and all the proteins which associated with flavin are commonly known as flavoproteins, like FMN, like FAD, like riboflavin, etc. Now, these are some of the common flavin molecules of plants. Riboflavin, this is commonly known as vitamin B2, and we must recall all the vitamins and their scientific names and their activities, abnormalities, etc. Anyway, the FMN or flavin mononucleotide, which actually derived from vitamin B2 or riboflavin, and the FAD or flavin adenine dinucleotide. This is a very active coenzyme associated with various proteins. Now, if we consider the cryptochrome mediated responses, we must understand these things, including stem elongation mechanism, leaf expansion mechanism, flowering, and circadian rhythms. Among this, some of the effects, to express some of the effects, the pigment cryptochrome work with other pigments like phytochrome. Now, this is the basic structure of the plant cryptochrome. Actually, these cryptochromes are composed of two domains: an amino-terminal domain or the N-terminal domain. This is known as PHR or photolyase homology region. And there is a C-terminal domain. This is known as DAS or domain of varying size. The PHR region of cryptochrome have two chromophores. One is known as pterine and the second one is known as FAD. Already we discussed about FAD. That is the flavin adenine dinucleotide. The pterine, like MTHF, that is the 5,10 methynyl tetrahydrofolate. 5,10 methynyl tetrahydrofolate. This is actually an example of a pterine. And these two parts act together to express the light sensory part. That is, the N-terminal region acts as a light sensory part and the C-terminal region acts as a portion which transduces the particular signal by interacting with other protein molecule known as E3 ubiquitin ligase, COP1, etc. So we must understand the basic structure of this plant cryptochrome. And actually, scientists studied this basic structure mostly on various types of model organisms. In this case, the model organism is known as Arabidopsis. And Arabidopsis thaliana, it is a member of the Brassicaceae or Cruciferae or the master family. They are commonly known as Drosophila of plant kingdom. As like Drosophila, which is a famous, widely available model organism from the plant kingdom, Arabidopsis thaliana also used as a very common model organism from the plant origin. Now, the plant cryptochrome's type. Normally they are of two types, like CRY1 and CRY2. But recently, a third type known as CRY3 also established. The CRY1, which is stable in light, but the CRY2 degraded in light. And this is the difference, but there are some similarity also, as CRY1 and CRY2, these two are both of nuclear origin, and CRY3 found in chloroplast, mitochondria, etc. And most interestingly, the CRY3 is completely different from CRY1 and CRY2, as it lacks a prominent C-terminal extension. So C-terminal extension is completely absent in CRY3. It is present in CRY1 and it is present in CRY2. Now, cryptochromes show its effect by blue light dependent phosphorylation activities, which results into subsequent conformational changes. And if we consider this thing, that phosphorylation and activation, they show similarity with phytochromes and other pigments also. And by this activation, there are some underlying changes or changes in the protein conformation, and this results into various associated physiological changes. Now, some important facets of cryptochrome. The activated FAD or flavin adenine dinucleotide chromophore path triggers the photoreceptor activation. So, this part, after the activation of this part, the cryptochrome shows its expression. CRY1 and CRY2 show different developmental consequences, obviously. Although they are of nuclear origin, but depending on their activities, on the presence of light, they show some differences. Cryptochrome also acts with the phytochrome pigments to regulate many developmental activities. They act together, like stem elongation or hypocotyl elongation, the flowering, circadian rhythms, etc. So, it has been found that the blue pigment cryptochrome acts with the pigment phytochrome in various situations to establish various photomorphogenetic expression. Thank you.